Thanks, ladies and gentlemen. Can you hear me?
Yes.
Okay, good. Most projects start with a question. They start with a problem. This one was quite interesting in that it started with the questions that weren't being asked. It's got quite a background, and we've been I've been sort of chipping away at it for probably about 10 years now. We have been chipping away at it now for about a year. And the discussion today is really also about people and people power and how getting people together can move an objective in the right direction. But I came into contact with a really dowdy little asclepiad called Brachysteelma natalensis about 10 years ago. I say dowdy, it takes a lot of loving. It's a fairly bland little plant. But a plant, nevertheless, that I've actually grown to really enjoy and love over the last couple of years. But we know that this little Brachysteelma natalensis there was one globally known population at that stage. There was roughly about 50 individual plants. That was all there was known to science globally, sitting in Kranskloof Nature Reserve. I was doing a bit of development assessment, and I got a phone call from a botanist the one evening who did the fault analysis on the property that was destined for development. So it was all about to be paved and trashed, to tell me they'd found 7 more specimens of Brachysteelma natalensis. So what this basically meant was this was the second globally known population that we knew of to science. And the question that wasn't being asked, which really irked me, was what are we doing to actually, we know we've now got about roughly sort of 57 odd plants here. How do we expand those plants? How do we ensure that this little thing goes off with a whimper and we lose a species we know actually nothing about? I'd like to get to know this plant before it's actually gone. And that's really how this thing sort of started, because when I started asking, the botanists, it was, well, how's your mind, dude? Put a fence around it. And I believe there needs to be something a little bit more active. So, hence the birth of our Ex-situ conservation project. And essentially, the 3 plants that we're concentrating on. The one is a critically endangered species, Brachysteelma natalensis. The other 2 are endangered. It's a little suffitex called Gymnosporia woodii, which was thought to have been extinct for about a century. James Medley Wood found a couple of specimens about a century ago, and Dave Stiles found a couple more on Ananda Mountain roughly about 10 years ago. As we've gone along, we've found we've got about 6 populations now, I think. But again, those populations, we've still got a lot of questions there. We'll look at that just now. And then the other one, which we also added to the list, was Delgrenodendron natalensis, a small tree. It's mainly noted from quite a few what do you call them? Specimens that were found at Umtumvuna. But we know of about 6 trees in the eThekwini municipal area, and that's an area of 2,200 square kilometers, 6 trees. So yeah, the first thing was to get a greater understanding of what such processes entail. We hadn't really sort of done this before. The breeding of rare and endangered species is nothing new. I think what is something which is fairly or comparatively new is that we want to put these things back into the environment. It's no good having a whole lot of rare and endangered plants sitting in a nursery. What ecological function do they fulfill? I remember a lecturer once said to me, he said, if you take no matter how rare that species is, you take it out of its natural environment, what is it worth? It may be worth something economically, but what is it worth ecologically? And I think that's really where we want to go with this. And a better understanding of the modelling process involved in identifying suitable and ecologically sustainable receiving environments. And Lyle at the back there is the brain boffin behind that. in terms of the modelling. We want to obviously go and find out where the historical areas where these plants originally were, and then add on things, some of those variables, like for instance climate change. As the climate changes, we're going to start seeing populations move. How do we model into that and find the best surrogate environments for the re-release of those plants? And then to gain a better understanding of the ecological attributes of these species. We actually know pretty much nothing about them. And what's been so exciting on this project is that we're picking up information all the time. It is empirical. This is not a scientific treatise. You won't find any graphs inside here or stats. But I'm hoping that there's enough impetus that it will interest more scientifically orientated studies at a later stage. Right, some of the objectives, and some of these were quite important. You know, we sat and we thought about where do we want to go with this thing. And I was always reminded of something that Roland Gutz said to me many years ago when he was with Museum Services. And they were in I think, Tsumeb, and they were collecting specimens for one in the museum of Cape, what they thought was normal Cape Griffin or Cape Vulture, only to discover at a later stage that this thing was a critically rare subspecies, and they'd probably wiped out about 2/3 of the globally known population group in collecting them. So I think the first rule of thumb is do no harm, and the last thing we want to do is run roughshod, go take propagation material only to find that we've actually decimated wild populations. So that was definitely something that was really important in this whole thing. Also to how do we store the stuff? How do we propagate it? Those are all the questions that we didn't really know at the start. But we've kind of done a little bit of experimentation. We've looked at what works and what doesn't work. And obviously, once these things go back into the wild, and I think that really, you know, for us is going to be the pinnacle of this project. And I've had a look at some of the research out there. There is a dearth of research, not a lot around. Probably the most interesting paper that I actually read was a Chinese paper. And every bit of information that I've looked at comes out with the same thing. It's very difficult often to re-establish wild populations, particularly when you're dealing with plants that are so specific. I mean, these things are probably on their natural extinction curve. We're not picking up that they've been heavily utilized for muti or for trade or for the horticultural industry or anything like that. But we have a moral obligation to keep these things going. And I think, you know, what they sort of saying is first of all, you've got to know something about the ecological, you know, what are these little things like and what don't they like? And we don't really know that information right now. We're hoping we're going to gain that information as we go along. And that will guide us in how we, you know, put these things back into the field. One of the little snippets that was interesting that came out of the Chinese study was that, you know, normally what you do is you take a whole of the seedlings and you plant them into an area. They're saying no, straddle it with a whole lot of different age groups. So plants with different levels of maturity, and it's a better safeguard of what you're putting back into the field. Obviously, that's now got to be, we've got to look at a long-term monitoring. You know, what goes back into the field, you know, what's going to happen there it's not going to help anybody. We plant these things back into an area, put a big fence around it and a 24-hour guard and spotlights and all the rest of it, and then don't bother to look at it again. We've got to monitor it. It's very important that we ensure what we've got expands. And once it expands, we can then, you know, hopefully start getting a lot more information about it. You know, the other thing was, particularly with things like the brachystoma, but also with the other two species. And, you know, just saying that, just going out of this slightly, you know, we're also toying and playing with the idea of maybe, you know, looking at some faunal species as well. You know, when we started this project, it was really to see, you know, can we actually do this? You know, we're a municipality. Do we have the resources in the municipality to do this? I would say at this stage, very definitely. But, you know, certainly with a lot of these plants, we need to know what's actually happening with them. You know, the brachysteuma hadn't been looked at for 10 years before we started looking for it. We went searching. I remember the day we went there, I think we found about 2 or 3 different species that we kind of thought was brachysteuma, and they turned into Thunbergia. And finally, Clint Carbot actually said, hey, there it is. Wow. You know, so, you know, nobody's looked at these things. Are they declining? Are they increasing? Are they stable? And then, you know, our methods of removing them, you know, are we doing harm? We don't want to do that. Okay. Challenges, we know very little about these things, but the really positive factor in this whole thing is, and I think I've got a bit of a freeze here. No, there we go. Has been who you team up with. But a little bit of that just now. I'm just going to run through the achievements because we've only got 5 minutes left, and I do dither around when I chat to people. Okay, the assegai population we haven't seen for years, at least a decade. There were originally 7 when Dave Stiles did his survey there. Clint Carbot and John Craigie went in, they found 3. I went back with one of our fundis, we found a 4th one. And that's really, really important, because one of the things which is also critically important, was on the last slide, is the heterogeneity of these species. The problem is that what we're doing is we're going to be creating clones. That's what you do with tissue culture. That's what you do when you take cuttings. It's a clone. And we want to try and avoid that genetic bottleneck. So it's really important that we've got more than one population so that we can keep that genetic diversity. But we've taken cuttings for micropropagation. Initially it was a problem because of contamination. Velocity Reddy, our able lady at our tissue culture lab, has tried out some methods of sterilization. We have had some contamination, but we've also had some really good results, particularly on the nodal cuttings. That, by the way, in the corner was Brachysteuma natalensis. Gymnospora woodea, very, very interesting. Poses a lot more questions than we have answers for. It's basically a little Suffratex. Up until a year ago, there was no known female plants. Nobody had found any female plants of the species. We only knew male plants. Where they grow, we know about 6 different colonies. We're getting about sort of 20 in a colony, but the critical question is, are we looking at 20 plants in that colony, or are we looking at 1 or 2 plants and a number of ramets? We didn't know that. So we did our own little quick sort of research, and it looks like what we're dealing with our ramets. We've also got some DNA ideas in the pipeline to see whether we can actually establish. We might be dealing with 5 plants, and that's all we know. But we see, we need to establish that. I'm almost there, ladies and gentlemen. The rest of the slideshow is actually just a lot of pretty pictures. Delgrenodendron, as I said, we know of 6 specimens. We found we found one that was in private ownership. The reason that's significant is that part of this whole project was it has to be obviously legally sound. And with this in private ownership, all we needed really was the landowner's consent to harvest, which we got his blessing. And we took a lot of cuttings from this plant, and initially it really, really looked good. We didn't do in vitro culture here. We bunged this in. We took tip cuttings, leaf cuttings, hardwood, semi-hardwood, and they were looking really good. The gymnosporias were looking really terrible when we first took the cuttings. That has, strangely enough, actually gone the other way. There is Velocity Reddy, our able boff there at, she runs our tissue culture lab, and she's been absolutely invaluable in this whole thing. That's Velocity's method of reducing contamination. She's basically using distilled water with a very small bit of chlorine in there. Everything's got to be really well timed to the nanosecond, and it's, as I say, we did initially have some problems. Those are the leaf cuttings. They were unfortunately contaminated. and we didn't get very far with that. Hands up, anybody seen Guardians of the Galaxy? It's a horrendous movie. Right, there's Groot, little guy that dances. Okay, now that was our first bit of really, that really excited us, because that's a lot of new shoots coming through. Those are our brachystelmas. There's nodal cutting, almost there. As you can see, there's the new shoot there, really great. The downfall of this is that we've got quite a lot of callus growths, so we're not quite getting the rooting right now, but we're working on that. And I'm going to just zip through some of the pretty pictures. We first used secateurs. We're now using sterile scalpels. That was to assess the damage. As you can see, that was the secateurs, very minimal damage, a little bit of chlorosis, and we want to go and see whether or not the scalpel's done any better. That's harvesting of our delgrenodendrons. Those are our initial cuttings. They look brilliant. We've got a cutting. That cutting looks still the same a year later. You're looking at the very, very first time any human being's ever seen a seed and a female plant of Gymnospora woodea. That's its seed. Unfortunately, they germinated, but they damped off. Initially, our Gymnosporas didn't do well. We have got some rooting. They're doing really well now. That's two. Well, it's actually one. There's two ramettes of Gymnosporia. And since we turned to what do you call it, root cuttings, we've had exceptional success. Thank you very much. I'm sorry, I think I may have gone a minute overboard, but does anybody have any questions?